In this video, we will discuss some of the results of the Tn-Antigen CAR T cells. So what does this mean? This means that this chimeric antigen receptor that this T cells contains is will target against this Tn-Antigen that are usually expressed whenever there is a change in the glycosylation profile occurs that leads to the tumor-like behaviors. And many solid tumors shows the change in the glycosylation profile, and in which this Tn antigen is more abundant, whereas in the case of normal healthy cells this Tn antigen is not expressed uh, or expressed in a very minimum level. And the Tn antigens, how this Tn antigen expressed? Tn antigen is usually the truncated form of the O glycan. So one of the reasons that this truncated form occurs is because of the epigenetic changes or mutation that occurs in the tumor cells that leads to the either we can say a mutation of the cosmic gene or epigenetic changes that leads to the suppression of the cosmic genes. And this cosmic genes helps in the stabilization of this O glycosylation process that will leads to complete O glycan profile, or we can say complete O glycosylation in the healthy cells, whereas in case of tumor cells this O glycosylation is not able to happen properly or processed properly because of the absence of cosmic gene that helps in the stabilization of the enzyme that finally helps in the O glycosylation. So because of that this truncated form of O glycosylation, that is Tn antigen, is more abundantly formed in the case of these tumor cells or solid tumor cells. So this researchers has developed this Tn antigen that can target only, or that can target or has the high specificity against this Tn that are expressed on the solid tumors. So in this experiment they checked their specificity, so whether there is a cross reactivity also occurs in this kind of anti-Tn antigen that are used in the CAR T cells, that will be used in the CAR T cells. First these experiments that results that are shown is only for this Tn antigen. Right now the results are not shown in this figure for these CAR T cells. So they have used different kinds of glycopeptides in order to check the specificity of this Tn antigen. So first one is this OSM. This is the heavily glycosylated glycopeptide that contains this STN part, and this OSM is we can say is the neuramidase treated OSM that does not contain the salidase part of this STN. And this MUC1 60mer, MUC1 60mer does not contains any of this glycosylated pipe. It is just the peptide without any glycans. The last one is the MUC1 9TN. It contains the heavily TN TN glycans in this within a short range of this MUC1 glycopeptide. So first they use the antibody, that is anti-T antibody, or they termed it as 3C9, and this is their negative control. And we can see it does not show any signals for all of the peptides, since none of the samples or glycopeptide contains this T antigen. All of them contain the Tn antigen. So then they did the check for this 5F4, which is the anti-Tn antigen that they have used further for the CAR T cell experiments. In this case we can see that the signals are high for this MUC19TN, and also there is some signal for this OSMTN. So this is in line with the rationale, or this is in line with the rationale or their expectation, as this 5F4 or anti-TN antigen will binds to the TN epitope that are present on the glycopeptides, whereas for other glycopeptides it does not show any significantly high signals. And next they use this anti-STN antibody. In this case they see the high signal only for this OSM without neuraminidase treatment, whereas they also see some signals for this OSM that are treated with neuraminidase. So one of the reasons could be that the neuraminidase treatment is not complete, or there are some glycopeptides of OSM that contains this silidase part. So its specificity, although it's good, but this region will show some kinds of non-specific binding. Next they check this 5E5 that has the specificity for both this TN and STN antigen. In this case they can see the signals are high for both this MUC91TN and also for this OSMTN, and simultaneously the results now, since the results are overlapping, we cannot see it, but the results are high for this MUC19TN, AOSMTN and OSMSTN. So this indicates that its specificity is high for this specificity for this 5E5 is also high, or its specificity is towards both the STN antigen and also the anti-TN antigen. And in this panel B we can see the schematic diagram of this 5F4 anti-TN antigen CAR, that is chimeric antigen receptor. This one is the variable light chain and variable heavy chain of this 5E5, or in other terms it is called as single chain fragment. And in the transmembrane domain they have this CD8 alpha, and in the intracellular domain they have this CD3 zeta and 4-1 BB. And we have understand this intracellular signaling in our previous videos. You can refer those videos in order to understand more about this intracellular signaling. Later on they also did the glycopeptide assay, in which they kept the glycopeptide in the cells and then add this different antibodies to check in which antibodies they can see in which antibody samples they can see the high production of the cytokines. So this is in other terms the indirect checking of this antibody binding to the epitope. So if the antibody binds to the epitope, what will happen is that they will release the cytokine signals, the T cells, or we can say the T cells. This is the T cells. So whenever this is the, you can say, chimeric antigen receptor, not just the receptor. So this chimeric antigen receptor, whenever it binds to the so this panel C represents the results for these CAR T cells. So this is the CAR T cells with the with the different antigens. So whenever these CAR T cells interact with the glycopeptide, it will show if it will release some cytokine signals. So the measurements are based on the supernatant data collected from the samples, in which for example these CAR T cells with Tn-Antigen interacted with the samples that contain some of the glycopeptides of one of this sample. So results are shown here. First panel C, first figure on the left side is about 5e5. So 5e5 has the specificity both for anti-Tn, anti-STN. So in this case we can see the signals are high for this OSM and also for this MUC19Tn. However, the signals are low for this neuraminidase treated OSM, and this is in line with our results from the figure A, and also in line with our expectations that 5e5 will binds to the STN and SN part. So, and next one is the result for the CD19 antibodies, CD19 CAR T cells. In CD19 CAR T cells, we do not see any signals of the cytokines in the supernatant, except for this OKT3. So this indicates that this OKT3 interacts with this CD19 CAR T cells. Next, they did the experiment with this 5e5 BZ cells. So in this case, 5e5 BZ cells, we can see the signals are high for more number of glycopeptides. That is, they are high for both the neuraminidase treated glycopeptide OSM and the neuraminidase non-treated glycopeptide OSM. So whereas the binding signals or cytokines signals in the supernatant are also seen for other samples such as MUC1 60mer and MUC19Tn. So this indicates that 5e5 has some kind of cross reactivity, or it might also react for the proteins that does not have this Tn and Tmer. And this CD19 BZ when it is has also shown. So one thing that I would like to clarify, this 5EB and CD19 results, the first two figures are for the interleukin 2 that is observed in the supernatant, and the second two results are for this IFN-G. So the CD19 also shows the signals for this IFN as similar to that of interleukin 2. So this panel C results indicates that this 5v5 has some kind of cross reactivity, whereas this CD19 interacts only with this OKT3 peptides. It does not have cross reactivity, but however it does not have the reactivity to target the cells that has the antigen that are present in the tumors. So it has some non-specific targeting of the CD19 cells. So this panel C indicates that most of the other samples or most of the other antigens has non-specificity. However, panel C results are not shown for this 5F4. 5F4. Yeah. So However, the results for the 5v5 shows that it does not have that much specificity, and if we use 5v5 antigen in the CAR T cells, it might lead to cross reactivity. So I hope you have understood something about how this something or the results about this anti-Tn-Antigen in the CAR T cells, and also the anti-Tn-Antigen specificity from the panel A. And if you need to understand more about this Tn-Antigen CAR T cells, you can go through the reference that has been provided in the description. That's all for this video. Thank you.